Ladies and gentlemen, a great pleasure to welcome to HKUST John Hardy, the man, the myth, and the legend. To make these comments as short as possible because I know that you're in for a very entertaining hour and a very informative one. A little bit of background. John received his PhD from Imperial College London. We'll skip the year. In dopamine and amino acid neuropharmacology, did a postdoc work at the MRC in neuropathogenesis in Newcastle, and then to the Swedish Brain Bank, where he actually started work on Alzheimer's disease. In 1985, he became, I wasn't going to do the date, he became assistant professor of biochemistry at St. Mary's Hospital in Imperial College London. And that's where his foundational studies in Alzheimer's disease really began. Fresh from the notoriety that those studies brought him, he moved to the University of South Florida in Tampa, then to the Mayo Clinic in Jacksonville, Florida, and where he became the chair of neuroscience. And then he moved from there to the National Institute on Aging at the NIH, and finally ended, his most recent stop in his peripatetic career is to have returned back to the United Kingdom to take up the chair of molecular biology and neurological disease at the Rita Lilla Weston Institute of Neurological Studies at University College London. He has won many prestigious awards, been recognized by many in the field as a true thought leader, not just in the genetics of Alzheimer's, which we'll hear about, in neurodegenerative disease, but also in the process of how you go about studying a human disease. So without any further ado or further words of introduction, please join me in welcoming John Hardy.
Thanks. It's a real pleasure to be here in this beautiful place. You know, I always love coming to Hong Kong. It's such a beautiful place, and I have to say the food is a lot better than English food too. It's really a pleasure to be here and to meet friends again. It's really nice to be here, and I hope to be coming back several times in the future. So I'm going to talk about neurodegenerative disease. I'm going to spend most of my talk talking about Alzheimer's disease, but at the end I'll talk about a few of the other major neurodegenerative diseases as well. These are the major, I think the major healthcare problems that are facing developed societies, as we have defeated largely, not completely of course, infectious disease, as we've controlled heart disease to some extent, and stroke through, if you like, blood pressure and cholesterol management and smoking cessation and so on. And as we are overcoming, to some extent, cancer, neurodegenerative disease is becoming increasingly important in our aging societies. And Alzheimer's disease is the major of those. It's about half of them. In the UK, where the population is about 60 million, something like 1 million people have Alzheimer's disease. You know, for that 1 million, there's another 1 million caregivers. So it's really a major problem for society. And I got into Alzheimer's research by accident, I have to say. I was hired to do work in Newcastle in Sweden on the pharmacology of Alzheimer's disease. And I'll talk a little bit about what we have known about the pharmacology and pathology. But in 1980, you know, when you study the pathology of the disease, which is how I started, what you're doing is you're looking at the end of the disease. You're looking at the car crash at the end of the disease. And in 1983, Gusella in Boston, working on Huntington's disease, located the gene which caused that disease to chromosome 4. And for me, that was really an important moment, because I suddenly realized that by studying the genetics of the disease, you could find where the disease began. So you could switch from looking at what the end of the disease was to seeing how it started. And in a simple-minded way, I thought if you could understand how a disease started as well as see how it ended, it would help you draw the path between and start to make sense of the biochemistry of the disease between. And it would be by understanding that biochemistry, that the pathway between the start and the end, that you might start to develop therapies. Those were my simple-minded ideas. And I was lucky enough to be hired to a department where a molecular genetics department. And so although at the time I was not a molecular geneticist, I had the opportunity to switch from looking at the pharmacology and pathology of the disease to looking at the genetics. So I was very fortunate in that way. So now what I'll do is I'll talk you through the genetics, first of all, of Alzheimer's disease and why that has led to attempts at therapy. I'll also talk about why I think those therapies have so far not worked. That's something that bothers all of us. And then I'll talk more about what I think about the disease in general, and then I'll move on to talk about other diseases. So that's my plan for the lecture. But incidentally, I'm going to leave all these slides here, so I'm going to leave them with Dr. Harrop, whose email address I'm sure you've all got, so you're welcome to the slides. You should not feel you have to copy anything down or anything like that. So this is the pathology of the disease, and what you can see here is several aspects of the disease. Here is a half brain at the top. This ventricle here, this space in the center of the brain, is far larger than it should be. And these sulci here are wider than they should be. There's been loss of neurons. There's been clear loss of neurons. And that's perhaps most evident here, where you see here in the hippocampal area, this is very atrophied. So this is what the gross histopathology, this is what the gross anatomy of the disease looks like. And here we're now looking at the three pathologic features of the disease. This is a plaque. This is about 1/10 of a millimeter across, perhaps a little less than that. And it's made up largely of a peptide, which I'll talk a lot about, called amyloid. And these are scattered throughout the cortical and hippocampal areas. Here we have a blood vessels, and these are surrounded by the same peptide, this amyloid peptide. So this is the amyloid of the disease. And then here, looking within cortical pyramidal neurons, you see these features here, which are called tangles, neurofibrillary tangles, and these are made up of a protein called tau. So this is what Alzheimer saw in his patient. And when pathologists saw this, they divided themselves into three groups. Those that thought the neurodegeneration was primary, and these were just, if you like, the tombstones of dying cells. Those that thought that this amyloid pathology was primary, and those that thought that these tangles were primary. That those were essentially the ideas. No one knew what caused any of this, but these were the, if you like, overarching ideas about what the order of pathology was, about what the relative order of pathology was. Now, most Alzheimer's disease occurs in individuals who are 65 plus. The incidence of the disease goes up from 65 to 70 to 75 to 80. And by the time you reach people who are 90, about half the people have Alzheimer's disease. However, there are rare families with early onset disease, and in those rare families, it behaves as an autosomal dominant disease. And that's what we started to look at. And this is the ancient history of this. This is the first family we worked out with Alzheimer's disease. And what you see here is a grandfather who was 55 years old. He had two sons who also are 55 when they developed the disease. It's not important for the story, but these two sons married two sisters here, and that's why the family tree is like that. And then this family had 10 children, 5 of whom got Alzheimer's disease. And this family, we collected 3 children, 1 of whom got Alzheimer's disease. And the experiment we did was incredibly simple. We said, what part of the genome is co-inherited with the disease? Which part of the genome do all the affected members inherit, which the unaffected members did not inherit? And what we found, and what you're looking at here, is the top half of chromosome 21, and you can see that all the affected members have got that part of the chromosome, but these two unaffected members this person has got the top of the chromosome but is not sick. This person has got the bottom of the chromosome but is not sick. So the simplest explanation is the disease gene is somewhere here and the amyloid gene was right in the middle of that section. And so what I felt I was doing there was behaving as the referee, saying there's all these competing ideas about what causes the disease, but we say that the genetics says that in this family, amyloid is what causes the disease. And that was really the simple process. Now, if I had a postdoc doing this experiment now, it would take 1 postdoc a year. No, much less than that. A good postdoc could do this in 2 or 3 months nowadays because of the technologies. At the time we did this, it took 6 of us 5 years to do it. So you're looking, this slide, nice and simple though it is, represents 30 person years of work. Nowadays it will be 1 month, 1 person month. And when we sequence the gene, you'll have never seen a sequencing gel like this, but this is the sequencing we did. And what you can see here is this is the affected individual, these 4 lanes, and this is the unaffected individual. And you can see there's a gap here, whereas in the affected individual, there's a heterozygous mutation, and that changes a valine to an isoleucine. And this was the first molecularly defined cause of Alzheimer's disease. And this led us to, if you like, in my view, it led me, if you like, to confirm the amyloid hypothesis. But this, in fact, is the first statement of the amyloid hypothesis. This paper from Glenner and Wong this work is 1991. This paper is 1984. What Glenner had done is he'd got the amino acid sequence of the amyloid peptide, and he got it first of all in Alzheimer's disease, but then he got it in Down syndrome. People with Down syndrome have got 3 copies of chromosome 21, whereas the general population have 2 copies. And he had said that since the sequence is the same, he wrote this, which I'll read. The cerebrovascular amyloid protein from a case of adult Down syndrome was isolated and purified. Amino acid sequence showed it to be homologous to that of the amyloid peptide of Down syndrome. This is the first chemical evidence between Down syndrome and Alzheimer's disease. It suggests Down syndrome may be a predictable model for Alzheimer's disease, assuming the name is changed, but assuming the amyloid peptide is a human gene product, it suggests the genetic defect in Alzheimer's disease is located on chromosome 21. So he says here that he thinks Down syndrome individuals get Alzheimer's disease because they make too much amyloid. That's really what he implies. And I regard really this is the first statement of the amyloid hypothesis. So what is so now I'm going to say where has genetics taken us to from that, and then I'm going to talk about why I think amyloid treatments have failed. And then I'll talk at the end about other things. So what am I saying here? I'll just say that. And what I should say is a lot of what I'm going to say in general is covered in this paper, which I published a few years ago, but also very nicely in this paper from a different perspective produced by the Cambridge group, looking at the proteomics of Alzheimer's disease and other neurodegenerative diseases. So this is what I've shown you. APP mutations are one cause of Alzheimer's disease. Peter Hislop in 1995 showed other early onset families with Alzheimer's disease had mutations in either presenilin 1 and presenilin 2. And then we showed in 1998 that you could have mutations in tau, but those mutations in tau did not give you Alzheimer's disease. In fact, they gave you a tangolony dementia, and I'll talk about that a little bit as well. But these findings overall allowed us to make mice through transgenesis with both plaques and tangles. And I know that, there are mice with these pathologies being bred here at this university. So these are, if you like, descendants of these findings. This is the amyloid hypothesis that we drew out. And it's incredible. Well, in fact, I would not say it's very simple. I would say it's overly simplistic. And I'm talk about why it's dangerously oversimplistic. So what it really says is here's APP, here's the amyloid gene. It leads to amyloid. This is the amyloid peptide. And that leads to tau dysfunction, tangles, and to cell death. And this is the process. So I started by saying that Gusella inspired me, if you like, to look for the beginning of the disease. I've shown you the end of the disease, and this is the pathway we drew to go from the beginning to the end. All sounds great. However, let's have a look at this diagram, which I drew in 1998. The first thing I will say is that there are these embarrassing question marks on the diagram. And, I wasn't embarrassed that they were on the diagram when we drew it in 1998, but actually I thought that we would fill in those question marks in the next couple of years. And it's now 2018, and those question marks are still there. And eventually you start to think that if you're asking a question and not getting an answer, maybe it's the question that's wrong. And, you should start thinking about whether you should ask other questions instead. The other thing I will say is that there's the implication here. It's not explicit, but there's the implication that this is the neuronal membrane, and here you have amyloid on the outside of a cell, and it's leading to tau dysfunction and tangles directly on the inside of the cell. In other words, everything that's important is happening with neurons. This is, if you like, a neuron-centric diagram. And I think that that's well, in fact, I wouldn't say it's misleading, it's just wrong. It is, we now know that is not the case. The other thing I think that I was wrong about, not wrong, but it's misleading, is when I was drawing this as a diagram, I was thinking of it as something like glycolysis. I was thinking that this was a pathway like glycolysis is a pathway. And of course, glycolysis is a pathway which operates over milliseconds. If you take glucose and then within a few seconds, probably much less than that, the glucose carbons are already in the TCA cycle. It's a pathway which operates over, probably milliseconds. That also is wrong. And what we now know is that this process is something which doesn't operate over seconds, it operates over years. And the idea that we should represent it in the same way as something that operates over milliseconds, I think is wrong. So I don't think that this diagram is wrong. I still, in a sense, believe in this diagram, but it is misleading. When it's misleading. That's what I would say, or you should not take it too literally. Anyway, so now I'm going to start and talk about amyloid APP processing, because that's an important component of the first part of this. And here you have the amyloid precursor protein here and here. And there are essentially two major pathways. The first pathway, which I'll call the good pathway, involves cleavage here by an enzyme alpha secretase just outside the membrane, and then a cleavage here. This is a rather complex event by the enzyme gamma secretase. Now, gamma secretase is in fact largely presenilin. The major component of gamma secretase is the protein presenilin, which is the other genetic cause of the disease. And this leads a fragment P3, so this is the good pathway. It does not lead to amyloid production. Here you have what I'll call the bad pathway. You have beta secretase cleavage first, and then gamma secretase, and here you have nasty amyloid. Those are the two pathways. I'll just say one important thing now. We don't know really what the function of APP is, and I think that's a major problem. We don't know if any of we certainly don't know if amyloid itself has a function, and I think that is also important. So these are important gaps in our knowledge, even though this has been studied for nearly 30 years. This is the pathway. Mutations in the amyloid gene cause disease and mutations in gamma secretase cause disease. That's old knowledge. This is what I've been telling you so far has been stuff that was done in the 90s. However, as we've gone now further in the last few years, we and others, and this is a paper from Rudy Tanzi, have shown that mutations, loss of function mutations in alpha secretase increase your risk of Alzheimer's disease. So if you have a loss of function in this pathway here, the good pathway, more goes down the bad pathway, so that increases your risk. So that is also consistent with the amyloid mutation, with the amyloid hypothesis. And then finally, a few years ago, the Icelandic group showed that a mutation in APP, which inhibited the bad pathway, also decreased your risk. So just going back to this diagram, a mutation here, which prevented this cleavage or reduced this cleavage, also, and therefore probably pushed stuff down the good pathway, decrease your risk of disease. So all of these things are consistent with the amyloid hypothesis. And then I've already talked about people with trisomy 21 inevitably develop Alzheimer's disease. There are these families who've got partial trisomies, and what you're looking at here is a gene dosage along chromosome 21, and you can see this family has got this part of the chromosome duplicated. They don't have Down syndrome. They have got the amyloid gene duplicated here, so they have 3 copies of the amyloid gene, and they too get Alzheimer's disease. There are people who have got this part of the chromosome duplicated, a few of them. We have a couple of them in our study, and those people have Down syndrome, but they don't develop Alzheimer's disease because they have not got 3 copies of chromosome 21. So all of this, all this genetic evidence totally fits with the amyloid hypothesis. All sounds great. Now, however, for the bad news, let's start going through the bad news. What can we do to use this knowledge to develop therapies? Well, one thing we could do is inhibit gamma secretase. I'll just remind you of the, so this is alpha secretase, this is beta secretase, and then this is gamma secretase at the bottom. So inhibit gamma secretase. Nope, patients got worse. This trial was stopped early because the patients got worse. That is pretty serious. I'll talk about that a bit later in a bit more detail. Inhibit beta secretase. No, this trial was stopped halfway through because the advisory board for the trial, who were looking in secret, if you like, at the data as it was coming out, said this drug, it's not making the patients worse, but it's not making the patients better. And no matter what the results of the trial, if we continue it, are going to be, it's not going to help, it's not going to be statistically useful. Immunize against amyloid and get and if you like, suck the amyloid out of the brain. This is actually a very complicated approach because everybody reacts to direct immunization slightly differently. So it's actually a rather difficult trial to interpret results from because everybody develops different antibody titers if you give and to different parts of the amyloid molecule. But this failed in phase 3. This failed in phase 3. Antibodies which target soluble amyloid and reduce the concentration of amyloid in the brain, and this failed a phase 3 trial. I'm a little ambivalent about saying this failed because, and you're not, if you don't ever say this to your student supervisor, the p-value was 0.7. You know, I think it was close. You're not allowed to say that. If you say it to your supervisor, they'll say, go and do some more experiments. But, you know, still, it was close. But it failed a phase 3 trial. And then antibodies which attack and remove plaques, there's actually now a couple of these in process, and they are still in phase 3. Not, though, a very good set of data, not though a very inspiring set of data. What's going wrong? Well, I think that what is going wrong relates to this, the time course. Remember, I said the amyloid hypothesis, as we drew it, looks as if it happens over milliseconds. But we actually now know, and in fact the data has been there for over 30 years in Down syndrome, that is not what happens. We know, and I'll just, that in fact, the disease starts much, much earlier. And here we have what are called the Jack curves. And what you're looking at here is age. Let's say this is somebody who gets to here, and they're 70 years old. And here they go to see their neurologist because their spouse, usually it's their spouse, is worried about them. And this is where they make the diagnosis of Alzheimer's disease. And let's say this is 70 years old. What we now know is that if we've been doing amyloid imaging in their brain, their amyloid in their would have started to build up in their brain somewhere about around here in their early 50s. And that if we was to look at their CSF amyloid, that would have started to change about the same time. Tau, that we'd have started to see evidence for tangles in their mid-50s to early 60s. And they would have started to show MRI and cognitive changes here, and certainly MRI change in their early 60s. So this disease process has been going on for a very long time. And now let's think about what those drugs were doing. Because the drugs which reduce soluble amyloid, and that includes the BACE inhibitors and some of the antibodies, those do not remove plaques. They just prevent further deposition. And they're being given here, in this period here. And of course, the amyloid is already right up here. And so you might be preventing further deposition, but it's already maxed out. So it's like taking a statin when you're having a heart attack. That's the analogy I would say. Statins might be the right drug, but this is the wrong time to take them. They're being given too late. The therapies are involved, those putative therapies prevent deposition, but they don't lead directly to removal. So this is why I think these are problematic. This is just what I've said. Now, with that in mind then, am I excited about drugs which remove amyloid from the brain? And I would say I'm interested, I'd like to be cautiously optimistic, but I don't think it's straightforward. Do we think that a brain from which amyloid has been removed is the same as a brain in which amyloid has never been deposited? We don't know that that's the case. It might be the case, but we don't know that that's the case. Is removing the amyloid ever going to be really beneficial? Obviously, one hopes so. Obviously, this company and other companies who have therapies which are based upon these antibodies hope so, but I don't think it's for sure. And it is actually the case that some people who had direct amyloid immunization came to autopsy, a few, there's only half a dozen, and this half dozen got amyloid immunization, so they were injected with the amyloid peptide. A proportion of them removed amyloid from the brain, and a proportion of those came to autopsy. So we're dealing with a proportion of a small number, so it's very small numbers. But these people, this small group of people, did have amyloid removal from the brain. They still had tangles, and they still died of dementia. And this is a quote from the paper. So one has to be, I think, cautious about that. Now, with that in mind, that's where we are up to with the trials. With that in mind, what's a geneticist to do? And the answer really is to try and understand what's causing late onset disease. These families are very rare. They're much less than 1% of cases. So we all started to study the genetics of late onset Alzheimer's disease. And these are the fruits of those labors. And this is genome-wide association studies. And what you do in genome-wide association studies, which were really invented in about 2005, what you do is using the gene chip technologies, you look at about 500,000 polymorphisms across the genome at once, and you test each of those 500,000 polymorphisms for association with disease, and ask the very simple question, are there any of those polymorphisms which occur more frequently in cases than controls? That's the statistical question. And this is pretty much where we're up to, and I'll explain the plot. Here you have the chromosomes, 1, 2, 3, 4, all the way down to 22. Then you have here the statistical significance. Now you're testing 500,000 polymorphisms, so you have to deal with that irritating Italian Bonferroni, and of course you're doing 500,000 tests, so you have to set your p-value very high. So you set your p-value at 10 to the minus 7, and this is the 10 to the minus 7 line here. Everything that goes above that line, you're allowed to declare as an Alzheimer gene. And here we have ApoE, which was discovered in the 90s by Alan Roses. This would go way through the ceiling. And then all these other genes here, we are allowed to declare as Alzheimer genes. And they have small risks. Now, the first paper to start to make sense of those of this is this paper from Leslie Jones, who pointed out that most of these genes fitted into actually 3 pathways. The innate immune system, which in the brain largely means microglia, endosomal vesicle recycling, which we think means APP processing. APP is processed through the endosomal vesicle system. We don't know that for sure. And what else? I've suddenly had a brain freeze. And lipid metabolism. So these are the three pathways that Leslie Jones pointed out. And the other point I'll say is that, in fact, not only do these genes largely fit to those pathways, but when we look here at these peaks which just fail Bonferroni, most of those genes are also in the same pathways. So they're probably true positives as opposed to false positives. So we can dig in a little bit more than that. And these are the first ones which were discovered just to illustrate Leslie's point. ApoE we'd known about for a long time, lipid metabolism, clustering lipid metabolism, and the innate immune system, PCAM, endosomal vesicle recycling, ABCA7, lipid metabolism, CR1, the innate immune system, BIN1, endosomal vesicle recycling, and MS4A innate immunity. So this and the other genes that we found subsequent to that fit this same pathway. Whoops. I just touched the screen. There we go, that's fine. Now, as we have looked at these genes, we have understood them more and more. And I'll just start by talking about this paper at the top from Alison Goet's group. And she pointed out not only that the gene is called SP11, but the protein is called PU1. And she pointed out that this was an AD gene, but that many of the other genes had this transcription factor were targets of this transcription factor. So she, if you like, integrated many of these findings by saying what you're looking at here is a microglial transcription factor, a genetic variability in a microglial transcription factor, and genetic variability in many of the targets of that transcription factor. So this is really an integrating piece of data. And we had done this experiment, which is described in this paper here, which I'm going to talk about now for a few minutes. And what we did in this experiment was we made mice with amyloid transgenes and tau transgenes, looked at their pathology as they got older, and at each age did whole genome expression using arrays firstly, but now transcriptome sequencing, and said what genes change in their expression during pathology development. And we found that in fact that this started to make sense in terms of the genetics of the disease. Now, we had been working on what I've been showing you is genome-wide expression work. We have been working also on sequencing, doing exome and genome sequencing of Alzheimer's disease, and we had found this gene called TREM2 was an important gene in Alzheimer's disease, and this is a microglial gene. And when we found mutations in this gene, we had already noted that, in fact, TREM2 was the gene whose expression changed the most during pathology development. So what you're looking at here is 2 totally separate experiments. Here, by sequencing, we had found TREM2 variants were important in Alzheimer's disease. And here, by looking at amyloid producing mice, we had found that TREM2 was the gene whose expression changed the most during pathology development. So two totally separate approaches had led to the same interesting gene. This is our data, and it's in a website called Mousiac, and here you have TREM2 expression going up as the mice age. As the mice age. So they're responding to amyloid deposition. And we did what's called whole genome, WGCA, whole genome, I can't remember what the other letters stand for. I will remember, but. So here you see, what you're looking at here in those mice is the genes whose expression alters. And here is, in the middle of this network is TREM2, and all of these other genes, their expression alters correlatively with TREM2. So TREM2 is the gene which alters the most, and all of these other genes are genes whose expression alters in the same way as TREM2. And interestingly, here we have 5 of the other GWAS hits for Alzheimer's disease. So we have found TREM2 is the gene whose expression alters the most, but within this little network, there are 5 other genes which are also GWAS hits for Alzheimer's disease. And not only that, but this was predictive because the next 2 genes which were described after our paper were ABI3 and PLCG2, and these also altered with TREM2 expression. So what we have just been doing, and we're just in the process of writing this up, is saying, what other are any of these genes also Alzheimer genes? So this is the question we wanted to ask, and what we did is there an over-representation in the TREM2 microglial network? And the answer to that was yes, there is, and the p-value for that was 10 to the minus 41. If we took out the genes that we already knew about, the p-value was still 10 to the minus 10. So this tells us that within that network, there are other genes which are also important in Alzheimer's pathogenesis. So which are those other genes which independently show association? And these underlined, this is the new network. Now we have switched from doing arrays to sequencing. Here's TREM2. These are the genes we already know about that had already been described. And underlined in blue are the new genes we are just writing up as being associated with disease. And here they are. These are genes we're just writing up now. And actually, you know, they are actually making sense. And nearly all of them have been discussed in relation to either microglia, in fact, with relation to microglia and Alzheimer's disease. OAS1 is involved in cytokine regulation. Gal3ST4 has been shown to be enriched in microglia clustering around plaques. CXCL10 is a pro-inflammatory cytokine. HLA had already been reported in the Finnish population as being associated with disease. This also has been described as being around plaques, as has this ITGAM as well. So we are seeing then, what we are showing here is genetic variability in amyloid response, in the response to amyloid, is important in Alzheimer's pathogenesis. So my conclusion here is that a response to amyloid deposition so genetic variability in the response to amyloid deposition is an important component of Alzheimer risk. However, how well you cope with amyloid deposition is important. And in fact, this fits really with in vivo work from Randy Bateman at WashU, who showed in other papers that young onset Alzheimer's disease were making too much amyloid, whereas late onset Alzheimer's disease, they were failing to clear amyloid. Now, the other aspect of this is where I'm going to go with this is we want to treat earlier, and I've already mentioned that in those Jack curves. But can we also use the genetic findings to predict who is going to get Alzheimer's disease? And the answer is, in Europeans, we are now pretty good. In Northern Europeans, we are now pretty good at doing this, because there's been all of these big GWAS in Northern Europeans, which allow us to predict. And the prediction accuracy is actually, well, in fact, it's actually about, the area under the curve is actually about 0.84. So in the general population, in Northern Europeans, we can be pretty confident who is at high risk just by doing a gene chip analysis and knowing their age and gender. We can be pretty accurate. We're never going to be perfect. We know we're never going to be perfect because identical twins are not always concordant with disease. Now that's in Northern Europeans. We're nowhere close to that with Asian populations because far fewer studies have been done. And this is one of the important questions that needs to be answered. And I hope that we here can be part of that. But we need to be able to predict who is going to get disease. And in Northern Europeans, we can do that. Now, what do I think? What I've been telling you so far is at least an approximation to the truth. Now I'm going to tell you what I think is going wrong. And what I think is going wrong is that, again, it is a failure of amyloid metabolism leading to membrane damage. And I will just go through this now. So what you're looking at here is the same APP processing. So this is the alpha secretase pathway, and this is the beta secretase pathway. You're getting amyloid production. It's going off into the extracellular space. However, what I think starts to happen to do with the metabolism of amyloid by gamma secretase here is you get a mismetabolism, and amyloid buildup starts in the membrane, and it's a very hydrophobic peptide, and starts to disrupt the neuronal membrane. And this disruption leads to amyloid deposition, and this is that period from when you are in your late 50s but not sick to when you start to get sick. You're starting to get amyloid deposition. But at this stage, what's happening is your microglia are coping with that deposition and cleaning up the mess. So this is the stage when this is the stage when amyloid deposition is occurring, but you're not sick. Essentially your microglia and other mechanisms probably are cleaning up amyloid, and you're not becoming demented. But eventually this process fails and is overcome. And it's interesting that all of these mutations, for example, the TREM2 mutation, which is a microglia, ABCA7, these are all loss of function. So these are all mutations which make the cleanup process less effective. And then this is the process which fails. This cleanup process is what eventually fails in late onset Alzheimer's disease. So I would say that late onset disease is largely a failure of damage cleanup, and this damage is amyloid-induced, largely amyloid-induced membrane damage, and that's what's going wrong. Now I'm now going to quickly talk about all the other late onset diseases very quickly. But the lesson I want us to take forward from now is that it's a failure. Late onset disease is a failure in damage repair, in my view, largely. And in Alzheimer's case, it's neuronal membrane damage repair. And I'll just now look very quickly across all of the other diseases. PD and DLB, what we're finding, these are the genes, pink, Parkin, pink, DJ1, FBOX07. These are all loss of function mutations which are in the mitophagy pathways. Mitophagy is the process by which damaged mitochondria are removed from the cell. So that one of the damage processes which is failing in Parkinson's disease is the damage repair, and it's mitochondrial damage. The other genes involved, LRRK2, synuclein, PL2G6, VPS35, these others, I won't name them all. All of these are involved in the endosome, lysosome, and the autophagy processes. And autophagy is a complex process with multiple functions, but one of the functions is to remove damaged cellular components, including mitochondria, from the cell. And so I would say in Parkinson's disease, the overall problem in late onset disease is mitophagy. Is removal of damaged cellular components through autophagy. And this paper is very persuasive. Genetic variability in genetic heterozygous loss of lysosomal enzymes is one of the risk factors in Parkinson's disease. So the loss, the damage that's failing to be repaired is probably intracellular damage to components, including to mitochondria. Then when we look at pure motor neuron disease, there's several pathways involved. Some of them are involved in axonal transport. I'm not going to talk about those. SOD1, I don't know what this is involved in, but all of the other pure ALS genes, FUS, Matrin3, TIA3, TDP43, they are involved in the stress response, in the RNA stress response pathway. They're all involved in the stress response pathway, and I'll just explain what this is from a simple-minded perspective. When a cell is put under stress, what it does is it changes its translational profile, and it does that by sequestering the messenger RNA in these stress granules, and then when the stress is removed, the stress granules unfold and allow the messenger RNA to be translated. And so this is a process by which the cell responds reversibly to stress. And these RNA proteins, TDP43, TIA1, Matrin3, and FUS are all involved in that process. So it's a failure in this stress response. When we look at the syndrome, which is both leads to ALS and FTD, what we find is that most of these genes, so these are unusual mutations. In some family members, they lead to motor neuron disease. In other family members, they lead to a dementia. We don't know why they have different phenotypes in different family members, but they do. And in these genes, certainly the majority, and probably all of them, are involved in the ubiquitin proteasome system. And the ubiquitin proteasome system is the mechanism by which damaged proteins are removed from cells. So again, I would say it's a failure. What we're seeing is that these late onset diseases are a failure, in this case, of the removal of damaged proteins. So you can see there's a large number of these genes all involved in the ubiquitin proteasome. And then finally, frontotemporal dementia, there's tau, which doesn't fit with what I'm going to say, but then there are these genes, progranulin and CHIMP2B, and GWAS hits too, that we and others have found. And these are all lysosome genes. So in pure FTD, what we're seeing is that a lot of the genetic, the importance, the problem is in lysosomes. So that it's a lysosome, which again I've just been saying is a response to damage is a way by which damaged organelles and so on are removed from the cells. So the overall principle I want to push forward is that late onset disease often is a failure of damage response. The gene complement we have when we're in our teens, 20s, 30s and 40s for removing damaged materials is okay. But as we get older, unfortunately, as those of you can see the skin of those of us who are elderly in the audience can see, as we build up, as we increase in age, we're less good at maintaining that youthful appearance. Our damage clear-up mechanisms are not working in our skin, and they're not working in our brain, and they begin to fail, and that's why we start to have neurodegenerative diseases. But the other point to say is these diseases, it's very interesting that each of these failures leads to different neuronal populations being killed. And so I think we're also seeing a clue as to selective vulnerability. I would say what we're seeing, for example, is that motor neurons need the stress granule response, and that's why if you have a problem with the stress granule response, it manifests first as a problem in motor neurons. And for each of these, I would say you're seeing a clue towards selective vulnerability. Different neurons, for reasons to do with their neuroanatomy and biochemistry, are susceptible to different insults. And, you know, I know that there's either none or only few clinicians in the audience, but I think it's really interesting that a patient comes into a neurologist, and what the neurologist says is not you've got motor neuron disease, but what he's implicitly saying is, I think you've got a problem in your stress granule response. So just by looking at a patient, I would say, to some extent, a clinician is making a molecular diagnosis of the underlying problem, in a biochemical problem in his or her patient. Thank you very much.
Really enjoy your talk. So for the failed clinical trial, you mentioned that perhaps the reason is because it is at an advanced stage of the disease, and so the clearing of the plaques may not have that much impact. I wonder if one also should consider, in addition to the plaque deposition, maybe there are other targets, for therapeutic treatment, so that in future maybe one can consider combinatorial treatment for the disease. So that's the first question. Second question, I actually would also like to know your view about models, because in the research lab, we all want to contribute to understanding the pathogenesis of the disease, and we use mouse models, genetically engineered mouse models, and more recently, the use of human iPSC-derived neuronal system. So I just want to see what you think about these models, and which one do you think would be a good model to study the disease?
What was your first question? You see there we have a problem.
The clinical trial.
Oh, yeah. I think Alzheimer's disease is a complicated disease. And I think, eventually we'll be using, many drugs at once in an individual patient. I don't think that we'll end up with a magic bullet ever. I don't think we'll ever end up with a magic bullet. We'll be taking multiple drugs. Now, you ask about other targets. One thing I absolutely think is very good science and clearly important is, for example, is the observation that tau pathology is self-propagating. And so there is a point at which, removing amyloid is past, is immaterial, because tangle pathology itself is self-propagating and is causing neuronal loss. So you're clearly going to need, I think, if you later in the disease, some anti-tau therapies. I do think understanding the damage response pathways and the microglial response to damage is going to be important, and that one can imagine ways of, if you like, pumping up the microglial response as well as reducing the amyloid damage. So you can either reduce the damage or increase the efficiency of the response, and I think that both are going to be important together, I think, with anti-tangle therapies. So I think eventually we will, and of course, as you well know better than I, there's synaptic damage, and that, what the relationship to that is not clear. Of course, microglia are involved in synaptic remodeling, and one can imagine that this is going to be a target, and we need to understand that process more, of course, to translate the limited knowledge we have now into therapies. So I would say, if I was to look 50 years away, I think we're going to be multiple therapies with multiple targets, with changing therapies during the disease. Yeah, that's where we're going, I would say, slowly. Models are models, and I think that the mouse models are very good models of amyloid deposition. And if you want to change amyloid deposition, I think the mouse models are very useful. I think that the more recent ones from Recon are better than the ones that we had before. But actually, although people say they haven't predicted outcomes, in terms of the pathology, they've been rather good at predicting utility. As behavioral models, they've not been so good. But in terms of understanding pathogenesis, I think they've been pretty good. And we're using stem cells and trying to develop 3D cultures. So we obviously believe in them. We haven't yet got anything out of them particularly, but we believe in them too.
So, Harley, that was a great talk. I have two questions. One, to follow up on the models. Recently, there are developed, the Jackson Labs developed models with risk variance as against traditional mutations. What do you think of their utility in studying this complex disease?
I think they're to be welcomed. One of the things we're trying to help the Jackson Labs with is taking all of the risk variants are very difficult. Risk variants, as your question implies, are difficult. If you put in a TREM2 mutation, that doesn't mean that that mouse is going to develop much Alzheimer pathology. What one can imagine is putting together, for example, the worst TREM2 mutation with the worst ABCA7 mutation and so on and so forth, and seeing how far down that pathway you can go. So I think that these Jackson Lab models are to be welcomed, and obviously with the CRISPR technologies we can get better at them. But I think, again, we should see them for what they are, which is models of processes rather than just a straightforward model of disease. We have had a tendency, which is just a kind of newspaper shorthand of saying these amyloid mutation mice are Alzheimer models. I say it all the time, it's a shorthand and it's misleading. They're models of amyloid deposition. And as long as we remember that type of thing, we'll be okay.
Okay, thank you. Can I ask another? Okay, great. Okay, so the polygenic risk score that you described, we've recently heard a lot of polygenic risk scores for complex diseases, cardiovascular, diabetes, etc. If it's really that simple, then can we use it now to screen individuals early on at age 50 and select them for imaging and catch the process? Is it ready for that?
I mean, within the bounds of what ethics committees will allow, yes. And also, unfortunately, we're in Northern Europeans only, because we haven't done enough work in Asians.
Okay.
So we have to get on with the work in Asians.
Yeah. Thank you.
And right at the back, there was one too. Farah, no, you go first. That's
Okay, so if I understand correctly, so you are saying that there's a damage response that has been failed in those disease, and then the protein that failed to be processed, and that's why they got deposited. So it sounds to me that if this is right, then the amyloid plaque is a byproduct of a failed system instead of a cause of the disease. Am I understanding that right?
Well, I don't want to get into a semantic argument. It's clearly not a good thing itself. I mean, it's a space-filling lesion. There's a horrible neuronal dystrophy around it, so it's clearly leading to damage around those neurons as well. So I don't want to get into a kind of semantic argument about whether it's a cause and effect particularly. I think that it's clearly a damaging lesion and it marks a damaging process. That's what I would say.
I see. Now, the reason I ask this is because I'm thinking that if that's the case, would we be a better strategy? Should we target the amyloid plaque itself, or should we target the process that actually lead to the plaque?
Well, I think the answer is both, but let me just say what I think is the thing that's most excited me about therapies in the last 2 years, and that is the therapy for spinal muscular atrophy. And in spinal muscular atrophy, you should look at the papers, not just, I'm going to give you a simple-minded view of it. By changing the protein which failed to be produced properly was SMN, and they changed the SMN splicing such that active protein was produced using antisense oligonucleotides. And that led to clinical efficacy. One of the ways which I think is entirely rational for Alzheimer therapy is to reduce APP production by antisense therapy, for example. So by that mechanism, you would be reducing the amount of protein and reducing the amount of damage. So that's one rational therapeutic approach. Or another rational therapeutic approach is to make the response to that damage more effective so that you remove the damaged protein more effectively. So I would say both are rational approaches, and both have merit.
Question in the back. How about if you just shout? So.
Okay, I'll shout too. It was a great talk. I love the overview of the genetics of Alzheimer's disease. And I was wondering if you can go into a little further, because you touched on it at the end about how all these different disorders, ALS, AD, they all have some common theme that's related to neurodegeneration. And my question gets at, because you touched on it a little earlier, about the difference between senescence. That's a common theme that I see among all these diseases. How do you differentiate between senescence and these diseases that makes it so unique for each one moving forward?
That's a very difficult question to answer straightforwardly, but let me just say, let's start with Alzheimer's disease. We all pretty much develop Alzheimer pathology. It was politically very important to define Alzheimer's disease as a disease. It was important politically because if we were going to persuade the US Congress to fund research into this disease, we had to call it a disease. If we were going to say we wanted to prevent the aging of the nervous system, we would never have got funding. So from a political perspective, we had to call it a disease. But we should remember that that was the motivation. I think of Alzheimer's disease as a process, and, the way that society is set up, we have this idea of disease, which really comes from the notion of infectious diseases, and really is not really directly applicable to something like Alzheimer's disease. But we apply it anyway because that's how the Federal Drug Administration is set up, and that's how society likes to think of these processes. But I think of them as processes and not dichotomous diseases. Now, I think that the example for Alzheimer's disease is the most clear that it's political. With the others, they're much rarer, and so it's much easier to conceptualize them as diseases, and therefore, if you like, different from the norm. But I would say the same basic principles apply to all of them. That as we age, our failure to clear damage becomes increasingly a problem.
All right, I'm going to do one as the chairman's prerogative. But using your analogy, if you don't take statins when you're having a heart attack, and my favorite analogy, doesn't help you to quit smoking once you have lung cancer, people will still get Alzheimer's disease and Parkinson's disease no matter what therapies we come up with. Where's the path to treating people who actually have whatever this process is? Is there still good biology to be done there? Is there still good work to be done there?
Yeah, I mean, the answer is yes. I mean, let's take an optimistic view of these amyloid therapies. Let's say they slow the rate of disease to a half of what the rate of progression is. Actually, what that's going to mean is there's a considerable increase in the number of people who are symptomatic and therefore will be at the moment taking cholinergic therapies. So the idea, we don't say this too loud to the politicians, because what we tell the politicians is we'll cure the disease. But what we'll actually do is certainly at the beginning, slow the rate of progression and increase the number of people with disease. And we don't tell that to politicians. I hope there's none in the audience. Because, that will really, they'll think, oh, I thought you guys told me this was going to save money. And in fact, in the short term, it's not going to save money. It's going to lead to increased valuable lifespan, but it's not going to save money in the short term. Don't tell it.
My lips are sealed. I think we've gone long enough, and it's been obviously a very stimulating morning. So please join me in thanking Dr. Hardy for